Welcome friends, welcome to another video from Channa's Biology, and in this video we will discuss about difference between the generalized transduction and specialized transduction. These are the both two types of transduction, different types of transduction, and there are some difference between these two types. So what is the main difference between the generalized transduction and specialized transduction? We will see in detail in this video. Okay, the first difference is that in generalized transduction the process is takes place by virulent bacteriophages. As you know that in generalized transduction the process is totally takes place with the help of bacteriophage, and the type of bacteriophage in the generalized transduction is virulent bacteriophage, focused on underlined virulent bacteriophage. Okay, while in specialized transduction, this transduction is processed by temperate bacteriophage, means that as in the generalized transduction, the virulent bacteriophage play an important role. In specialized transduction, the transduction is takes place with the help of temperate phages. Okay. Now the second point is that in generalized transduction undergoes lytic cycles, mean generalized transduction follows the lytic cycle, while as specialized transduction follows the lysogenic cycle. Okay. In the third point, any portion of gene from whole DNA of donor bacterial cell is transferred. As you know that, as name indicates, generalized transduction. In generalized transduction, from the whole DNA of donor bacterial cell, any portion of bacterial gene, there is a no restriction. So it is also known as unrestricted generalized transduction. Any portion of bacterial gene can be transferred from one bacterial cell to the another bacterial cell. While in the specialized transduction, only the restricted or only the specialized genes from the whole DNA of the donor bacterial cell is transferred to the another recipient bacterial cell. Okay. The fourth difference is that in generalized transduction, virulent DNA is not integrated into the bacterial DNA or bacterial chromosome. As you know that in generalized transduction, the process is totally takes place with the help of virulent bacteriophages, and the virulent bacteriophages containing virulent DNA are always, remember that they are not incorporated, they are not integrated with the bacterial DNA or bacterial chromosome. But while in the specialized transduction, the viral DNA is integrates with the bacterial DNA. Viral DNA is forms the integration, is forms the complex is along with the bacterial DNA. The fifth difference is that donor bacterial DNA is packed inside the bacteriophage in the generalized transduction. We know that in generalized transduction there is a one phase is that, and in this the transduced phase is formed, and in this transduced phase the donor bacterial DNA is packed as a individually. Okay, but while in the specialized transduction, along with the donor bacterial DNA, small part, small fragment of the virulent DNA is attached with it, okay, inside the bacteriophage. Means that in specialized transduction, the donor bacterial DNA is not present as individually as single. It is forms. It is present along with the virulent DNA. Okay. Sixth difference is that in generalized transduction, bacterial DNA hydrolyzes into pieces by the virus. Okay. In generalized transduction, due to the viral DNA or due to the virus, the bacterial DNA is hydrolyzed into pieces. It is break down into the small fragments, small pieces. But in specialized transduction, bacterial DNA is not hydrolyzed, while instead the bacterial DNA is incorporated, integrated with the virulent DNA. Okay, so this is the totally difference between the generalized transduction, specialized transduction. If you like the video, then press the like button and hit the subscribe button for the more upcoming videos. Thank you very much.